I'll do it as I usually do, since it's just mostly reminders and nothing new. So I'll go ahead and get started early with that. So hopefully we can start the actual lecture at 8 a.m. So anyway, again, it is too late to retake exam 1 to change your grade, but you can still keep retaking it for practice because your final exam is going to consist of about 100 questions, and about 50 of them are going to come from the first 3 exams. So at this point, exam 1 is now basically a study guide for the final exam. And also, you can meet me online to get all the correct answers for exam 1 and a 5% boost to the grade. And of course, I'm not just going to give you the answers. I'll also talk to you and explain why your answer is wrong and why this is the right answer and help you remember it. So it'll benefit you in 2 ways. Again, not only do you get the correct answers, but you get a 5% boost to your grade. Exam 2, you can still retake it. You can just like exam 1, you can retake it as many times as you want. It's open book. It's not timed. Just make sure you let me know when you're done, because I have no way of knowing that you've done that unless you tell me. And then once you do tell me, I'll go in there and I'll average your first grade with your highest grade. And there's nothing to lose because, again, like I said, well, first of all, no one got that high of a grade. So especially for exam 2, there's nothing to lose. I can't imagine how you could do any worse. But if somehow you did do worse on the retake and you keep retaking it, and you still can't do better than you did the first time, then we'll just keep your first original grade. So again, nothing to lose. And that is open until exam 3 is due. And I'm not sure when exam 3 will be due yet. We'll have to play that by ear. All depends on when we finish the exam 3 materials. So any questions about exams 1, exam 2, or for that matter, exam 3? Okay, this is a reminder, the genetics lab that we did this past week is due today. Well, past due today. And the pre-lab for the DNA lab is due today as well. Normally I say prioritize the pre-lab, but today, in this case, it doesn't really matter. Because next week's lab is 100% virtual. So we might be meeting, just so I can be here if you need my help. But we will not be working as a team at all next week. Everybody's gonna be doing their own individual little thing. So because of that, I don't really care. I mean, you still need to do the pre-lab, but it's less important than the previous weeks because, you don't have classmates depending on your understanding what's going on. So any questions about what's due today, the lab and pre-lab? Okay, on that note, I'll remind you I'm here to help, right? You don't have to do any of these things alone. If you have any questions about any questions about the lab or the pre-lab or anything else for that matter, let me help you. My office hours on every Friday are 9 to 10, so I'll be online and in my office from 9 to 10. But you can reach me anytime, well, not anytime, you can reach me after that as well. So just a reminder, I'm here to help. Also, usually on the weekends, I like to prioritize time with my family. So now's the time to get your work done, right? So if you need help, I will work on your pre-lab and your lab today. That way, if you need me, you can reach me and I'm there to help you. Which isn't to say don't contact me on the weekends or at night. Like I said on day 1, you can contact me anytime you want. Call me, email me, anytime, any day. If I'm asleep, I'm gonna turn my ringer off so you'll never bother me. So yeah, contact me anytime you need to. I'm here to help. What else? And then attendance. As usual, attendance is gonna be held the same way, or done the same way. I'm gonna get a series of attendance words throughout this lecture. You're gonna write them down. If you're watching the video at a later date, you're gonna take a screenshot every time I say one of the attendance words. And then when it's all over, send me an email with the list of words and one question about the material covered in the video or lecture. But of course, also, again, if you're watching the video, in the email, include the screenshots. Just like I did last time, for those of you who are in person, however many words I give out today, you're only going to need 1 less than that. That way you can get a little extra credit. So again, for example, if I give out 4 attendance words and you send in 3, that'll be full credit. Meanwhile, if I give out 4 attendance words and you send in all 4, you'll get a little extra credit to your participation class. So let's try to do that more often. So any questions? This is usually the same old stuff, really nothing new. But I have had some people say, hey, I didn't remember the due dates. So I'm reminding you once again, every Friday, labs and pre-labs are due. So any questions? All right, we'll go ahead and take attendance very quickly, and then we'll get started. Ashley is not here. Barrett is here. Wooten is not here. Trahan is not here. Dickens is not here. Clark is not here. Inca is not here. Isaiah is not here. Copes, Smith is here. Smith is here. Watson is not here. Kaylee is here. Lucas is here. Marley is not here. Noah is here. Savannah is here. Mia is here. Edwards is here. Wilkes is not here. Did I miss anybody? All right. So, we'll get started. Let's see here. Where you at? There we go. And the first word for attendance today is going to be spring. Since today is the first day of spring. Good morning. All right. So let's jump back into it. We had just finished giving the looking at the big picture of meiosis, right? We looked at the big picture. Now we're going to dive in and look at each individual stage. And to remind you, for the most part, these questions about meiosis are going to be more comparing and contrasting the differences between meiosis versus mitosis. There'll also be some comparing and contrasting between meiosis 1 versus meiosis 2. So keep that in the back of your mind as we go through this, because, well, that's basically how the questions are going to go. So here we go, prophase 1. Prophase 1 is essentially the same as the prophase that you learned in mitosis, with some differences, and you're definitely going to need to know the differences. First of all, for all of meiosis 1, not just prophase 1, but for all of meiosis 1, we're dealing with homologous chromosomes, right? We're not even dealing with sister chromatids yet. We're dealing with those homologous chromosomes. It's a very important difference. And again, that's not just for prophase 1. That's all of meiosis 1. Homologous chromosomes only. If you see any questions about mitosis and meiosis that deals with homologous chromosomes, you should automatically think meiosis, specifically meiosis 1. Also, like I said, we're dealing with homologous chromosomes. So if you remember in mitosis, we had these sister chromatids that were attached together, and they were attached together at the centromere, and we called them sister chromatids. And that's still happening in the background, but we're not talking about it right now. But what I'm getting at here is when we have these homologous chromosomes, those are also paired together. Because if you remember, one of the things we talked about on Wednesday was the fact that we have crossing over, right? These homologous chromosomes are right next to each other, right? So you've got your mom, the chromosome you got from your mom, the chromosome you got from your dad, they're basically attached right now. And that pairing, what we call that a synapsis. I'm not sure if I'm going to use that word or ask you about that on the exam, but I'm definitely going to use that word in the lecture. And I'm definitely going to use the word in the exam, so you will need to know what it is. So a synapsis. And again, you could almost compare a synapsis to talking about sister chromatids, right? So remember, sister chromatids is when you have that one chromosome that's been duplicated and they're attached to each other. Here we have the pair of chromosomes attached to each other in something called synapsis. Like we've already mentioned previously, but it's very important, so I'm going to say it again. Prophase 1 is when crossing over occurs. And also, like I mentioned with mitosis, your textbook goes a little bit deeper than I'm going to go for the exam. So your textbook doesn't just talk about prophase 1, it also talks about prometaphase 1. Just so you know, I'm combining it all into one to make it easier. Also, I have a little note here to tell you. Your textbook also mentions something called a chiasmata, a recombinant sister chromatid, and tetrads. So if you want to, if you're a note taker, you might want to write that down. Your book covers that, but I'm not going to. I'm not going to cover it. There will be no questions about it on the exam. If you read it in the textbook, and for some reason, I shouldn't say it that way. If you're reading it in the textbook and you're interested in it and don't understand it, then let me know, and we can meet and I can explain it to you. But you don't need to understand it for the exam, and you don't need to understand it for anything that comes after this chapter. So that reason, for this 100 level course, I'm not including it. So any questions about Prophase 1? All right. This picture's in the wrong spot, so let's get past that. Just like in mitosis, right after prophase comes metaphase. In this case, we're talking about metaphase 1. This is the first metaphase of the first meiosis. Right? It's essentially the same as mitosis, except, again, we're dealing with homologous chromosomes, not sister chromatids. So still, we have things meeting in the middle, just like we did in mitosis and metaphase. But now the things that are meeting in the middle, they're not sister chromatids. They're homologous chromosomes. And this is an important concept, and it's going to get a little bit more complicated later down the line. For now, it might seem simple, but later it will get more complicated. This bullet point will be very important. The arrangements of these chromosomes as they meet in the middle, these homologous chromosomes, are random. So you know the shape of a chromosome, but I'm not going to do it that way. I'm going to do it like this. Imagine you have 3 chromosomes. So you've got your mom chromosome and your dad chromosome. Let's call that chromosome number 1. They paired together. I'll try to draw a cell. I'm so bad at drawing. Here's a cell. They're paired together in the middle. Here you have chromosome number 2. You got your mom chromosome and your dad chromosome. They paired up. They met in the middle. Then you got a third one. Mom chromosome, dad chromosome. Let's say this species, whatever it is, only has 3 pairs of chromosomes, 6 chromosomes. This is metaphase 1. They've all met in the middle. Now, the way I drew it is the way you just might think of it. Like, all right, mom's chromosomes are on one side, dad's chromosomes on the other. But what I'm telling you in this bullet point is that it could happen that way, but it's random. So it could be mom, dad, mom, or mom, dad, dad, mom, or dad, mom, dad, like all these different ways, right? That's going to be very important a little bit later, and this is called independent assortment. So I'm going to take a little time before I move forward because it's that important. And once we really do start talking about independent assortment, like I said, it's going to get a little bit more complicated. So I want you to be very aware of what independent assortment is. They don't always line up the way I drew it. As a matter of fact, statistically, that would be very rare. It's all random. It's not all one, mom chromosomes on one side, dad chromosomes on the other. It's completely random. Any questions about this slide? All right. Once again, I'm going to go to the next slide, which is a slide that is out of order because we're talking about meiosis 1, specifically metaphase of meiosis 1. But the next slide is another image of meiosis 2. So sorry about that. I don't know how that happened. All right. So hopefully you already know what comes after metaphase, whether we're talking about mitosis or meiosis 1 or meiosis 2, what always comes after metaphase is anaphase. In this case, this is called anaphase 1 because it's the first version of anaphase, the first round of anaphase. And again, you should see a theme here. Anaphase 1 is essentially the same, and telophase are essentially the same as everything you learned about mitosis, except once again, we're talking about homologous chromosomes, not sister chromatids. So in anaphase, it's the homologous chromosomes that have finally been pulled apart, as opposed to sister chromatids. In telophase, it's the homologous chromosomes that are now on the opposite ends of the cell, not the sister chromatids. And what you don't need to know, and you can look into this for extra credit if you want, and I don't remember if your textbook mentions it or not. So remember in telophase and mitosis, that was the end of it, right? So everything had to go back to normal. In telophase and mitosis, the chromosomes uncoil, right? Because they don't need to be coiled up anymore. The nuclear envelope reforms because the job's done, so we need those things in the nucleus. The mitotic spindle disappears because we no longer need to move them around, so we don't need anything attached to them. All that stuff happens in telophase of mitosis. So my extra credit question for you, if you want to look into it, and again, I don't remember your textbook might tell you, but if it does tell you, you do not need to know this. This is just extra information if you want to look into it. Does all that other stuff happen in telophase 1? Because think about it. If we're talking about meiosis 1, this is only the first round of meiosis, so it still needs to go through another round of meiosis. So do we want to reestablish that nuclear envelope just to have it disappear again? Do we want to detach all the mitotic spindles just to have them reattach again? Do we want to uncoil the chromosomes just to have them coil up again? So that's things you can look into if you want. But again, for the exam, I'm not going to get that deep. You definitely need to know what happens in telophase of mitosis. The only thing you need to know about telophase of meiosis 1 is that we're dealing with homologous chromosomes as opposed to sister chromatids. Any questions about that side? All right, you guys are quiet. Here we go. Finally, now we're getting into meiosis 2, right? We're done with meiosis 1. Notice another thing I didn't mention, because I'm not going to get into it on the exam. But cytokinesis, I didn't even mention that. But yes, cytokinesis definitely happened. Back up the slide. So I should have written that down. I don't know why I didn't. So yes, we have anaphase 1 and telophase. Meanwhile, as you learn from mitosis, cytokinesis is happening. And we know it's happening because remember, in the first round of meiosis, we started with 1 cell and we end up with 2 cells. Now, these 2 cells are going into meiosis 2. And again, there's going to be a lot of comparing and contrasting on the exam. So these are the kind of things you need to understand for the exam. So meiosis 2 as a whole is essentially the same as mitosis, right? We're still dealing with, now we're finally dealing with sister chromatids again, just like we are in mitosis. But there are some differences. For example, the end product is going to be a cell with half the genetic material, aka it's going to be haploid, right? So again, to remind you, because this will be a test question. In mitosis, you start with 1 diploid cell and you end with 2 diploid cells. In meiosis, you start with 1 diploid cell and you end up with 4 haploid cells. Combine these 2 bullet points right there, right? Also, let me rephrase that, because there's different ways of saying this. You're going to need to know them all for the exam. In meiosis 1, you start with 1 cell, in mitosis, you start with 1 cell, and you end up with 2 genetically identical diploid cells. In meiosis, you start with 1 cell, you end up with 4 genetically unique haploid cells. That doesn't sound like that would be that hard of a question, but it is. Usually, multiple choice questions are easier than other questions, but in this case, it could be trickier. Just think about all the different combinations I can have to throw you off the correct answer, right? So the correct answer, again, if I said, what is the result of meiosis 2, meiosis, the correct answer would be 4 genetically unique haploid cells. But there's all kinds of versions, the different ways I can change that to make it incorrect. So I could say meiosis ends with 2 genetically unique haploid cells. And if you're just glancing over that, that would seem correct. But then you'd have to look closely and say, oh, it's not supposed to be 2, it's supposed to be 4. I could say 4 genetically unique diploid cells, right? And that would be mostly correct, but the fact that I said diploid would make it incorrect. So again, I'm just telling you now that question can be tricky for that reason, but it's not a trick question, which is why I'm bringing this up now. Let's see. What are we talking about? Homologous pairs of chromosomes and other. Oh yeah. So all those things I was telling you about earlier, there's just pictures of it right there. So what we're looking at here, remember the whole purpose of meiosis 1, which we just finished talking about, was to split up the homologous chromosomes, which is what this shows right here. Right? Shows, and this is from your textbook. It just shows us breaking up those homologous chromosomes. And then in meiosis 2 and mitosis, now we are splitting those sister chromatids. So any questions about this slide? All right. The next word for attendance is fall. Because today is the first day of fall, if you're in the southern hemisphere. So if you guys have any friends down under or whatever, they are now starting fall. All right, so here's a picture I've been showing you on accident, but here's now the proper time to show you. Here's all of meiosis 2 in one picture. So again, we've ended meiosis 1. Imagine if meiosis 1 was over here we have one cell, and then it's, it goes through all the prophase, metaphase, anaphase, telophase, cytokinesis, and then it ends up in two cells. Now these two cells just do exactly what we do in mitosis, right? We have these sister chromatids. You can see, well, eventually they're going to get attached to the mitotic spindle. The nuclear envelope is gone. They're coiled up. They're going to move towards the middle of the cell. They've met in the middle of the cell. That's what we call a metaphase. Right now, what we can't see is there's a tug of war trying to pull apart those sister chromatids. Eventually, when one of them is pulled apart, we call that anaphase. Now, at this point, they're being pulled farther apart, and the cell is getting an oblong shape. Finally, they make it to the opposite ends of the cell, which is what we call telophase, right? The mitotic spindle disappears. The nuclear envelope reforms, which you can't see there, and the chromosomes uncoil, which you also cannot see there. But again, mitosis 2 is basically the same as, meiosis 2, basically the same as mitosis. Other than the fact, again, like we said on the previous slide, we end up with 4, even though you can only see 2 here, but you can see all 4. They just haven't, they're split, but they haven't been separated. But there's 1, 2, 3, 4, right? We have 4 genetically unique haploid cells. So any questions about this slide? All right, now to bring it all back together, we're going to have a quick review comparing mitosis and meiosis. Because again, for the exam, a lot of the questions about meiosis are going to be comparing and contrasting the two. So for both mitosis and meiosis, we're dealing with a chromosome duplication that happens only once. So remember, the chromosomes duplicate in S phase of interphase, and that's true for both of these. And again, it only happens once for both of these. So it's not like it goes through meiosis 1, and then it goes through the whole cell cycle again and duplicates again. It doesn't do that, right? Once it goes through meiosis 1, it goes directly into meiosis 2. Cell divisions are where it's different though, right? In mitosis, like we've already said, we only have one cell division, meaning they duplicated once in S phase, so everything doubled, all the genetic material doubled, and then it was cut in half in mitosis, bringing it back down to the original number. Therefore, it is diploid, right? We started with one cell, we now have 2 genetically identical diploid cells. Meiosis is different, right? We do the one duplication, so however much genetic material we have, we double it, right? Then we cut it in half in the first round of meiosis, so we're now back down to the original number. Then we cut it in half again, so now we're down to half the original number of chromosomes, right? Half the genetic material. But also 4 haploid cells. So again, started with one cell, one diploid cell. We ended up with 4 genetically unique haploid cells in meiosis. So this is all, there's no new information here. I'm just bringing it together on one slide so you can see it a different way. So any questions about this review of a slide? All right, so some more comparing and contrasting. Here we go. The events that are unique to meiosis occur in meiosis 1. Another way of saying that is essentially, other than the differences we've already talked about, meiosis 2 is basically the same as mitosis, at least for us on this 100 level course. Obviously, if you were to really look into it, you would find some other differences. But for the most part, meiosis 2 is virtually identical to mitosis. Again, the key thing that at least for the exam, is with meiosis 1, we're dealing with homologous chromosomes. With meiosis 2, we're dealing with sister chromatids. And again, unlike in mitosis, meiosis 2 yields haploid daughter cells. So they're not diploid, they are haploid. And again, I don't know why I don't have this written, because this is a really big, important concept too, is independent assortment, right? So independent assortment happens in meiosis. And to remind you, independent assortment means when all those homologous chromosomes line up in the middle of the cell, it's not like it's all mom on one side and dad on the other. It's a random combination. So if you were to look at any of your sperm cells, well, I'll give you the numbers later, but if you were to look at your sperm cells or your egg cells, each one of them statistically should have a different combination. Like the way your mom's chromosomes and your dad's chromosomes have lined up in the middle of metaphase 1 should be completely different. Every single cell you look at statistically should be different than the next. That's what makes you so unique. And again, I'll give you some numbers later. That's when it'll get even crazier. But are there any questions about this slide before I give like a visual comparison between mitosis and meiosis? All right, here it is, mitosis and meiosis, visualized. This is a picture that's from from the old textbook. So if you find this one easier to follow than the pictures that are in your textbook, you have access to these PowerPoints on WVSU Online, so I suggest using them. But here we go, right? Mitosis versus meiosis. Here, in prophase, we're dealing with sister chromatids. Then in prophase 1, we're dealing with homologous chromosomes. In prophase 1, we're dealing with crossing over. We have crossing over. In prophase of mitosis, that does not happen. Metaphase of mitosis, we have the sister chromatids that lined up in the middle of the cell. In metaphase 1, right, it's not, they're still lining up in the middle of the cell, but they're not the sister chromatids yet. We still have to separate these homologous chromosomes. So that's what's lined up in the middle of the cell. And of course, in anaphase and telophase of mitosis, we end up with two genetically identical diploid cells. And by genetically identical, I mean identical to themselves and the parent cell. And then, of course, when we're talking about meiosis, anaphase 1 and anaphase 2, that's only the end of the first half of the story. Then it goes through another round of meiosis, and again, we end up with 4 genetically unique haploid cells. And really quickly, even though this is a very simplified version, we're looking at a species here, or an example, that only has 2 pairs of chromosomes. There's chromosome 1 and chromosome 2. Think about us, we've got 23 pairs of chromosomes, so it's going to be a lot more complicated for us. But even with this simple example, here you can see this sperm's, or this gamete has, I don't know, let's just call red the mom's chromosomes, right? So this one's got chromosome number 1 is completely from mom. Chromosome number 2 is mostly from mom, with a little chunk of dad on it, thanks to crossing over. This other gamete has chromosome number 1 that is mostly from mom, with a little chunk of dad, and chromosome number 2 is completely from mom. This other cell has chromosome number 1 that is completely from dad, and chromosome number 2 that is mostly from dad with a chunk of mom on it. And then finally, the 4th cell has a big chromosome number 1 that is mostly dad with a chunk of mom. And then finally, chromosome number 2 that is completely mom's. So right there you go. Just looking at something that has only 2 pairs of chromosomes, you can see that it's completely different. So again, imagine how complicated it is for humans who have 23 pairs of chromosomes. Which brings us to this, the origins of genetic variation. This is very important. So yes, we are discussing, this is obviously important to this chapter, which is why it's in here, but it's also very important to things you're going to learn later. For example, next, even though we've already done it in lab, after this, we're going to get into genetics, basically. To truly understand genetics, you need to understand independent assortment, which is what we've already talked about, but now we're about to go into it in greater detail. To understand natural selection, which will be later in the semester as well, you can't truly understand natural selection unless you understand this idea of genetic variation. Because without genetic variation, you don't have natural selection. And for that matter, you wouldn't have any form of evolution. Not just natural selection, but any type of evolution. So anyway, we're going to discuss this. We'll talk about the origins of genetic variation. And then if we have time, we'll talk about what happens when things go wrong with meiosis. Just like we did last chapter, right? We talked about what happens when things go wrong with mitosis. We talked about cancer, things like that. But now we're going to talk about what happens when things go wrong with meiosis. All right. So the offspring of sexual reproduction are genetically different from the parents and one another. I've already said that. I'm saying it again. It's a very important concept to understand and to know for the exam. Right? When we're talking about sexual reproduction, the offspring are unique. They're different from the parents. They're different from each other. Again, you're going to learn later on when we talk about evolution and natural selection. That's going to be a very important concept. So the question is, how does meiosis produce this genetic variation? The answer is we've already talked about it. But now we're about to talk about it in greater detail. And I don't think your textbook mentions it, so I'm going to go ahead and mention it, even though we're not going to talk about evolution until much later. There are evolutionary advantages to sexual selection versus asexual selection. So for example, if you're an organism that lives in a very stable environment that's not changing much, if at all. Like for thousands of years, your environment isn't changing. And maybe you're an organism that doesn't even get to see other organisms that often. Asexual reproduction would be great for you, right? First of all, asexual doesn't require another partner. So if you're, let's call it, I don't know, maybe you're some kind of amoeba that lives in a cave. How about that? That's a good example. So you're living in a cave. You're not going to see other amoebas to mate with. You're living in a cave, so things aren't changing much, right? The water level is not going to change much. The lighting is not going to change at all. The temperature, the ecosystem, the things that are living in there, it's a very stable environment. So if you have evolved to thrive in that environment, then it makes sense to make a perfect copy of you, right? Like you are perfectly adapted to this. So why not make a perfect copy of you? Because you're perfect. Sexual selection, on the other hand, is different, right? Sexual selection, we have these varied offspring. I mean, obviously the offspring are similar to themselves and similar to the parents, but they are different, right? And that's useful when you have a changing environment. Some of those offspring are going to have a slight advantage over some of the other ones. And that's going to be useful because as time changes and the environment changes, you're going to need something to be stronger than the rest because that's going to be your next breeding stock. Therefore, the next generation will be stronger than the next. In a changing environment, you don't want a perfect copy of yourself. Anyway, that being said, let's talk about how meiosis produces genetic variation. Again, we've already talked about both of these things, but we're going to talk about them again specifically. Independent assortment, we've already talked about it, right? As you already know, this happens in metaphase 1. Right? In metaphase 1, those homologous chromosomes line up together, unlike this, not sister chromatids, but the homologous chromosomes. Meaning, again, when you look at all of them, for humans, all 23 pairs are lined up. And sometimes it's going to be mom on one side and dad on the other, right? And it's completely random. And I told you earlier I was going to give you some numbers, so here I am, I'm giving you some numbers. Since we have 23 pairs of chromosomes, that means there's about 8 million possible combinations of the way your chromosomes can line up. Meaning, like I was saying earlier, right? I said if you were to look at your sperm and egg statistically, if each one of them would have a different combination of mom on which side, dad on the other. Specifically 8 million. Think about that. That means if you were to look at 8 million of your sperm, not even considering crossing over, but if you were to look at 8 million of your sperm or eggs, statistically, each one of them should be different than the other. Regarding all right, chromosome 1, did he get mom's version or dad's version? Chromosome number 2, did he get mom's version, did he get dad's version? Chromosome number 3, did he get mom's version or dad's version, right? And again, that doesn't even include crossing over, right? So even if you could somehow find 2 gametes, sperm or egg cells in your body that are the same, as far as that's concerned, mom on one side, dad on the other. Even if you could find that, which would be a 1 in 8 million possibility, then there's crossing over. So even though you might say, oh, look at that, both of them have chromosome number 1 from mom. Yeah, but maybe one of them's got chromosome number 1 from mom with a huge chunk of dad on it. And maybe the other one's got chromosome number 1 from mom with a little tiny chunk of dad on it. Right? So it's when you put those numbers in there, I don't think anybody's put numbers to that. So anyway, 8 million is a big number, but then consider that, it's even bigger. So what that means, and there's some more math here, if it's 8 million possible combinations for one sperm or egg, that means it's also 8 million possibilities for the other sperm or egg. So the probabilities of that specific sperm, that specific arrangements of chromosomes, meeting with that other egg with that specific arrangements of chromosomes is 1 in 64 trillion. That means, other than things like identical twins, which we won't even get into that, has nothing to do with this. That means your parents could technically, in theory, have 64 trillion kids, and statistically, you should be different than each one of them. Not one of you would have the same set of chromosomes. And again, this still doesn't even include crossing over. And I know this is where it gets a little bit I'll say risque, but bordering inappropriate for a conversation academically, but I'm gonna mention it anyway. Think about the possibility of like what it must have taken for you to be you. Your parents had sex to create you, assuming that's how it happened. It wasn't IVF. Like my son was born from IVF. Even then, the numbers are staggering. But think about it. If there's 64 trillion possible combinations, your mom and dad had to do it the exact same way at the exact right time. Does that make sense? Like nothing could have changed. If your mom had a cramp and had to lay like this instead of like this, any tiny little thing would change and you wouldn't be you most likely. The idea of that sperm combining with that egg is 1 in 64 trillion. It's a mind-blowing idea. Not to mention, each human on Earth has that staggering statistics. Then there's you, yeah, but your mom being your mom was that rare, and your dad being your dad was that rare, and then so on and so forth. You know, go back generations. It's just incredible to think about. So the math might be boring. Math usually is to most people, but to me, that's just incredible to think about. Like if your dad or mom got off work a little bit later than they should have, and then didn't get to it a little, you know, exactly when they did, you wouldn't be you. You would be a different person. Anyway, any questions about this? That might be a number you should know, by the way. Sorry. I get so interested in this as a topic that I forget we're talking academically. So yeah, maybe you should know those numbers for the exam. Not that one. Whoops, excuse me. 8 million. 8 million possible combinations for your gametes. Therefore, 64 trillion combinations for the fertilization. Anyway, any questions about that? Okay. Next word for attendance is ravioli. Today is National Ravioli Day. Don't know why. Anyway, so here we are talking about these huge astronomical numbers. Here's just a quick visualization from the old textbook of all the different possibilities from a species that only has 2 pairs of chromosomes as opposed to us, we have 23. So again, and I've already shown you this earlier, basically it's the same thing I showed you earlier. With this combination, this species that only has 2 chromosomes, there's only 2 possible combinations. There's all mom on one side and all dad on the other, or mom on one side for that chromosome, dad on that side for the chromosome, right? But again, think about how complicated that could be, or how complicated it is for humans. Anyway, any questions about that? Okay, so like I said, those huge astronomical numbers don't even include crossing over, right? So again, even in that 1 in 64 trillion possibility that sperm from your mom and that sperm from your dad came together, or a sperm with that arrangement of chromosomes from your mom, and good grief, an egg with that arrangement of chromosomes from your mom coming together with a sperm with that arrangement of chromosomes from your dad, right? That was this 1 in 64 trillion chance. But then think about the crossing over. So even if you did somehow have 2 sperm and egg like that, then there's crossing over. That just throws those numbers right out the window. Anyway, so again, as you already know, crossing over is the exchange of corresponding segments between homologous chromosomes, right? So again, we have 23 pairs of chromosomes. And what I'm saying there is when in prophase 1, all those 23 pairs of chromosomes are coming together. So the version you got from your mom is right next to the version you got from your dad for all 23 pairs of them. And because they're identical in that they have the same genes on them in the same location, sometimes they literally just swap chunks. And this results in gametes, egg and sperm, that have chromosomes that are partially from the mother and partially from the father. And if you remember from what I said, well, I'll bring it up on the next slide, and it'll be easier for me to show you than to talk about it. So the next slide I'm going to show you just a visualization of crossing over. You've already seen visualizations before. This next one's not it's not particularly better or worse. It's just a different one. And again, there's no new information on this slide. I'm just reviewing things I've already told you before. Crossing over happens in prophase 1. It's when we're just swapping chunks of homologous chromosomes. But anyway, here's a picture of it. Unless everybody needs me to slow down. Really good? All right. Anyway, here's where you can see it happening, right? So we have these homologous chromosomes that are next to each other. Something happens. We don't even get into what happens. You can look into it for extra credit if you want. It almost looks like they're cuddling up to each other. And in a sense, you might be able to say that. But anyway, then they swap chunks. And I like this picture because the last picture I showed you, they only swapped one chunk. In this picture, they swapped two chunks. But just like the first time we brought this up, like I told you, sometimes it doesn't happen at all. Sometimes it happens, with only one chunk. Sometimes it happens with two chunks. Sometimes the chunks are really tiny. Sometimes the chunks are really big. So it's completely random. So again, that 64 trillion, which is a huge number, is basically just astronomical when you consider this too, because it would be essentially impossible to get two of the same, this sperm and that egg coming together to create you. That'd be darn near impossible. Now, granted, if it was a really small chunk, a chromosome, at the very tip, that's probably not going to make much of a difference, but still. Any questions about this slide? All right, here's where things get slightly more interesting or more important as far as the exam is concerned. Let's talk about what happens when meiosis goes wrong, right? What happens when things don't work like they're supposed to? Because again, like when we talked about with mitosis, when things didn't work out that way, you ended up with cancer. Things are different here. What can happen with errors in meiosis? Long story short, and of course we're going to make it long again, but long story short, it can result in genetic abnormalities ranging from mild to severe to fatal. In other words, what can happen when meiosis goes wrong? You can have the wrong number of chromosomes, and that could be, barely make a difference, or it could kill you, or anywhere in between. That's the big picture. Know that before you start learning all these other details. That's the biggest thing to know, right? If meiosis goes wrong, you end up with the wrong number of chromosomes. And we've already talked about this a little bit, so we're bringing it back, right? You can visualize this with a karyotype, as you did in, what was the last lab? Yeah, not the lab we did this week, but the week before that. You've already seen the karyotype. You were asked to look at karyotype and find the problem, find the chromosome pair that had the wrong number. And then you just had to look it up and, tell me the name and the prognosis of that condition. But anyway, any questions about this slide, or can we jump into it? All right. First of all, let's talk about how the accidents during meiosis can alter the chromosome number. So I already told you the result is going to be the wrong chromosome number. Now let's talk about how that happens. The answer is something called non-disjunction. Know that name, know that word for the exam. Non-disjunction. This is where the chromosome pair fails to separate at anaphase. Of course, the end of that sentence is kind of redundant. Technically, you could just say it's when the chromosome pairs fail to separate. Right? Obviously, it's at anaphase because that's when the chromosomes separate. And when this happens, when any pair of chromosomes fail to separate, that will then obviously produce gametes that have an abnormal number of chromosomes. I'm going to give you some visualizations a little bit later to show you some examples. But yeah, obviously, all the pairs of chromosomes are supposed to separate meiosis. And then let's just say one of them doesn't. You can just think, right? If one of them doesn't, then obviously one cell is going to have too many chromosomes and the other one's going to have not enough chromosomes. And that's basically it in a nutshell. Of course, obviously we're going to dig deeper, but in a nutshell, that's the big picture. Another thing to know is that it can happen during meiosis 1 or meiosis 2. And the next bullet point, and I haven't been read for this reason, because the next bullet point is basically saying the same thing in a different way. So when we're talking about these chromosome pairs not separating, they could be either homologous chromosomes not separating, or it could be sister chromatids not separating. Hopefully you can see how that's the same, right? Those two bullet points are basically the same. Because homologous chromosomes not separating would be meiosis 1. Sister chromatids not separating would be meiosis 2. One thing I don't think your textbook brings up, and you definitely don't need to know because this is a 100 level biology class, but think about this. If we're talking about chromosomes not separating, Logically, that can also happen during mitosis. So my question to you, if you want to look it up for extra credit, is what happens when you have this issue with mitosis? Obviously, it's a bigger deal here because in meiosis, the whole purpose is to produce a new organism, right? We're producing sperm and we're producing egg. So that's a problem because, if you start a new life, right? If you have that zygote and it has the wrong number of chromosomes, then that's it forever, right? Because you all started as one cell and then you went to mitosis and became 2 cells and 4 cells and 8 cells and 16 cells. But each one of those is a perfect copy of the first. So you can see why it's a bigger deal and why we talk about it during meiosis, right? If you have a messed up sperm or a messed up egg, then the resulting human being, in this case, all the cells will be messed up. So look it up if you want for mitosis, but long story short, we know it's not that big of a deal because the mitosis, like if you had that happened in some skin cell in your arm, you might be affected right there, but it's not going to you're not creating another human being from your arm. So it's not that big of a deal, which is why your textbook doesn't mention it, I don't think. Anyway, any questions about this slide? Okay, so like I said, the big picture before we get into the details, what happens when something goes wrong with meiosis and mitosis? It's something called non-disjunction, meaning the chromosomes don't separate like they should, which gives us the wrong number of chromosomes. But now there's words for that, just to make things more complicated for biology students. So if you have the correct number of chromosomes, that is called euploid. Whether you're talking about you as an entire human being or more likely if you're talking about a cell, that cell has the correct number of chromosomes, that is called euploid. Now, if it does not have the correct number of chromosomes, that is called aneuploid. It includes monosomy, which is the loss of one chromosome, right? Mono being one. Trisomy, which means you have an extra chromosome, right? So for humans, we're supposed to have 2 of each chromosome. So mono means you have 1, you're missing 1. Trisomy, 3, so you have 1 extra. I think that's all the examples your books gives, but it actually gets bigger than that. Plants, for example, even though you don't need to know this, plants actually do quite well if they have 4. If they have double the chromosomes, sometimes that's actually better for the plant. Plants are different than animals. So yeah, what we're talking about right now, what we're about to talk about, all these conditions and how they're good or bad, talking about humans. So again, for extra credit, I invite you to look into other species. Are other animals like this? You just bracket up, right? What about other mammals? What about other vertebrates? What about other non-vertebrate animals, like sponges, jellyfish, spiders, whatever? Then you start looking at plants, fungi, protists. How does this stuff affect those other organisms? Anyway, I'm going to show you a picture to give some examples of some non-disjunction. Before I do, does anybody have any questions about these terms that you might need to know for the exam? All right. So here we have, like I said, non-disjunction can happen during meiosis 1, which is where the homologous chromosomes fail to separate, or it can happen in meiosis 2, which is where the sister chromatids fail to separate, because again, remember, it's basically like mitosis. And this shows both possible outcomes, right? So if it happens in meiosis 1, what you end up with is still 4 cells, right? Because meiosis, you start with 1 cell, you end with 4. Here you can see this gamete has too many chromosomes, this gamete has too many chromosomes, this gamete doesn't have enough chromosomes, this gamete doesn't have enough chromosomes. In other words, if non-disjunction happens in meiosis 1, it's worse than meiosis 2 because all 4 of your gametes are not right, so to speak, right? We see here if it happens in non-disjunction, if non-disjunction happens in meiosis 2, here we have 1 cell that has too many chromosomes, 1 cell that has not enough chromosomes, and then 2 normal chromosomes. 2 cells with normal sets of chromosomes. So that might be a nice, easy test question. Which would be worse? Non-disjunction in meiosis 1 or non-disjunction in meiosis 2? The answer is non-disjunction in meiosis 1 would be worse than non-disjunction in meiosis 2. Because again, when it happens here, all 4 of those gametes, those sperm or those eggs, they have the wrong number of chromosomes. It's half as bad in the other situation. So any questions about this slide? All right, so the next slide I'm going to show you shows an abnormal gamete fertilizing a normal gamete. And in the example I'm about to give you, it's going to result in a zygote that has 2N plus 1 chromosomes, meaning it has the full normal set of chromosomes, which is what the 2N means, plus 1 extra chromosome. And then, like I said earlier, the reason this is so bad with meiosis versus mitosis is because once that's fertilized and you have that one zygote, that one copy, that one cell is going to be copied and copied and copied and copied, and that's what the new organism is. So if it's one cell, that one starting cell is messed up, all the resulting cells will be messed up. So if the organism survives, which it does not always, then it'll have an abnormal karyotype and a syndrome of disorders caused by the abnormal number of genes. So all the stuff you looked at from last week's lab. And again, for extra credit, I invite you to look up other stuff, right? So you learned about trisomy 13, trisomy 18, you learned about an extra X chromosome, a missing X chromosome, an extra Y chromosome, maybe 2 extra Y chromosomes. I don't remember everything you learned, but if you want to for extra credit, you could look into some other examples. Like what happens if this chromosome has too many? Because we didn't do all 23 chromosomes in that lab, right? There was only a handful. Anyway, here's the picture I was talking about. This is from the old textbook. You can look at it if you want on PowerPoint. Here we have a normal sperm cell, right? It's got 2 different chromosomes. We'll call it chromosome number 1, chromosome number 2. This one from the mom has 2 chromosome number 1s and 1 chromosome number 2. So when they come together, we have a full set of chromosome number 2. We have a full set of chromosome number 1. But we also now have an extra chromosome. And again, more credit ideas. Why is that so bad? Like, how is it that having extra genes is so detrimental? Like, I could see it's a bit common sense will tell you like why having 1 fewer would be bad, right? Because you need those genes. So why is it that having extra chromosomes, i.e. extra genes, why is that bad for humans? Anyway, here's the book example. You've already learned about it in the lab I keep talking about. This is the most common example, so this is why we're going to talk about it. I probably won't ask you any questions about it on the exam, but here we go. Trisomy 21, right? This is where you have 3 number 21 chromosomes as opposed to what you're supposed to have, which is just 2. Now, if you're looking at the whole genome, that means you have 47 chromosomes as opposed to 46. A person with trisomy 21 is said to have Down syndrome, and this affects about 1 in 850 children. It is the most common chromosome number abnormality, which is why the textbook probably mentioned it. It's also the most common serious birth defect in the U.S. Again, which is why the textbook probably mentioned it. And the risks increase with the age of the mother. So the older the mother gets, the more likely it is. Specifically, when a woman is 35 years older, they are then candidates for prenatal screenings. I put it next to that just to indicate visually that I'm not going to ask you any of this on the exam. The textbook presented the information, so did I. But again, there's a lot of stuff you can look up for extra credit if you want to. First of all, are these numbers still accurate? I don't remember when this was published. Is it still just 1 in 850 children? Is it still the most common chromosome number abnormality? Is it still the most common serious birth defect in the U.S.? Also, are there other countries where it's the most common birth defect? Also, what are some of the other most common birth defects in other countries? Yeah, that part should still be true, but you can still look into it. Obviously, the risk does still increase. Or why? That's a good question. Why? What is it about women getting older that causes this to happen? Why not men? Right? Why don't sperm carry the same kind of risk? Your book also points out that people with Down syndrome have characteristic facial features. They very often, but not always, have a shorter lifespan. There's varying degrees of developmental delays, and some of them may live to be the middle age and beyond. Once again, I just presented the information because your textbook did. I'm not going to ask you anything about that. I invite you to look any of that stuff up, to look into it more for extra credit. Here's a normal karyotype. You can see here, chromosome number 21 looks normal. It only has 2 pairs. Here's somebody with trisomy 21. You can see there's 3 pairs of chromosomes. So that's it. It's going to be weird. So when we get back,